Hi everybody, welcome back. In today's video we're going to talk about Chapter 5 in the Snustad genetics textbook entitled The Chromosomal Basis of Mendelism. Um, Up until this point we've been reading about sort of the uh, output, the observable phenotypes that Mendel and others worked out. We talked about classic Mendelian genetics, and then in the previous chapter, chapter 4, we talked about sort of the extensions, they called it, to Mendelism, or these sort of variations on what can happen when things don't behave in a strictly Mendelian fashion. But this chapter sort of pivots from genetics as an observable phenomenon to tie that observation to something cytological. So, people had been looking at cells for quite some time. Um, by this point, into the early 1900s now, they knew cells existed. We were observing how pieces of the cell could move, microscopes were in use, all those things. And so the drive in the early 1900s was to sort of figure out what part of this cell was contributing to this genetic inheritance. The Mendelian stuff, the linkage between genotype and phenotype was reasonably well understood by that point. Of course, we figured out more things since then, but the title of this chapter, The Chromosomal Basis of Mendelism, really gets to this point that the work described in this chapter is talking about how to link the Mendelian processes to a physical part of the cell, which of course we know is DNA on a chromosome. So you can see the chapter outline here on the screen. We're going to talk about chromosomes themselves, the chromosome theory of heredity, and then focus in on, this is the new genetic part for us in this chapter, is talking about sex linkage. Um, Pretty much everything that we've looked at so far, we have assumed that there were two alleles for each thing, um, for each gene. So in the Mendelian case, right, we had two alleles of a gene, and those would be inherited independently. Now, sex-linked genes are those where the gene encoding a particular, protein or process, that gene is located physically on a sex chromosome. In humans, the X or the Y chromosome. And the only reason that that's different, why that poses a little bit of a challenge for us, is that males and females, as you hopefully know, have different composition of those sex chromosomes. And so someone who has only, say, one copy of an X chromosome would have a different phenotype for an X-linked gene than someone who has two copies of that chromosome. And so we'll get into that. This chapter focuses a lot on the fruit fly. You'll see references to a couple of important scientists right here on the first page. We see Thomas Hunt Morgan, sort of has been called the father of chromosomal genetics in some places. Morgan's lab really was a powerhouse in establishing a lot of the rules we're going to talk about in this chapter. And so now on to the slides. The slides are available, of course. You can follow the link there in the description. But what we're looking at here really is that outline that was presented in the text. And so just to remind you of some chromosomal vocabulary, these are probably not new words, at least I hope they're not. But chromatin is our sort of broad term, which could encompass euchromatin or heterochromatin. There are two varieties here, reflecting whether that chromatin is expressed or not, the content of that genetic material is expressed or not. And then on the right side of the screen, we have the language that we're going to use to talk about how many copies of the genome a given organism has. And so the word here, haploid, or N, would refer to having one copy of the genetic material. Some organisms are haploid for their entire life. So you could talk about, say, an E. coli cell. It has one copy of the genome, and that's it. So when you talk about making mutations in an E. coli cell in the lab, you only have to alter one copy of the DNA, and you see the phenotype that results. The only time in which a bacterial cell might have two copies is during cellular reproduction, during that process of mitosis. But generally speaking, in the course of its functional vegetative lifespan, it's a haploid cell. Um, Diploid is common amongst animals, at least. Lots of animals are diploid, humans included. You'll see a lot of the problems in the textbook at the end of the chapters sort of are diploid centric, written by humans for humans about things like humans. So if a problem is asking you something about expression patterns or something like that, and it doesn't explicitly specify the ploidy of the system, um, you can assume diploid. This is the case where the organism, the adult organism, has two copies of each gene, two full copies of the genome. In humans, every one of our somatic cells has two copies of the genome. And so when we talk about the human genome being represented across 23 chromosomes, that would be our haploid number. We have 23 physical pieces of DNA that make up our entire genome. But every adult cell has two copies of each of those. And so we end up with 46 total pieces of DNA in our cell's nucleus. In the case of humans and many other organisms, when we go through sexual reproduction, then our diploid cells, through the process of meiosis, you can refer back to chapter 2, form haploid gametes. We call them egg and sperm cells in humans. In other organisms, they might have different terms, but those gametes have half the normal complement, one copy of the entire genome. And then when they fuse together in the process of fertilization, we reconstitute a diploid copy. There are organisms, particularly when we get into the plant kingdom, there are lots and lots of higher ploidy organisms. So tetraploid and octaploid are very common. Hexaploid, I didn't write on the slide, that'd be 6 copies. This is the case in the same way as diploid, where now the adult cell has multiple copies, 4 or 6 or 8 copies of the entire genome. If that organism goes through sexual reproduction, which of course many plants do, their gametes would have half their normal complement. So a tetraploid plant would generate diploid gametes, each of which then have 2 complete copies of the genome. When those gametes fuse back together, pollen and egg, for instance, we now would regenerate a tetraploid organism. The same is true with higher ploidies, octaploid, 8N in the normal cells, 4N in each of the gametes. The key thing maybe to realize here is that when we talk about model organisms, there is obviously a significant difference if you're trying to induce a mutation or you're trying to observe a particularly recessive mutation in an organism. The fewer copies of that gene you have to work with, the more easily observable that trait would be. So in the yeast world that I worked in, yeast can exist in either a haploid state or a diploid state. So it's kind of a cool organism in that respect, in that it can undergo both types of growth pattern. In the haploid state, it's very easy to make a mutant and to observe them. In the diploid state, you can get some dominant and recessive masking. And so we typically would then sporulate or cause the diploid yeast to go to a haploid form so that we can identify those mutant alleles without being masked. It gets harder, of course, if you're dealing with a tetraploid or a hexaploid or an octaploid organism. If you're trying to delete a gene, for instance, in a tetraploid, you would have to make that deletion 4 separate times in order to get a completely null mutant. Here is a table from the text just showing you really a lot of data, and to my mind, it's not particularly obvious what the organization here is, but I've highlighted a couple of important species that you might want to know about. What's shown on the right in this column is the haploid number. This is the number of physical pieces of DNA in the cell nucleus that represent one copy of the genome. So it's not necessarily saying anything about the size of the genome. What it is saying is how many physical pieces of DNA have to be accounted for during processes like mitosis and meiosis, and DNA replication, that kind of thing. So when we look at, for instance, 2 different fungi up here at the top, Baker's yeast, that I mentioned a moment ago, has a haploid number of 16. That means in that haploid yeast strain that I talked about, there would be 16 pieces of DNA floating around in the nucleus that represent all of the genes of the yeast. In the diploid strain, there would be 2 copies of each of the 16 pieces, and so 32 total pieces, but really think about it as 16 pairs. However, when we talk about Neurospora, this common bread mold, it has its DNA, its genome organized over only 7 pieces. We can see as we go down here, Arabidopsis is a common plant. It's a little mustard green that lots and lots of plant labs use. That thing has 5 chromosomes. It often comes in a tetraploid format, and so although the genome is split over 5 pieces, there are 4 copies of each, and so there's 20 pieces of DNA floating around in there. Drosophila, we'll see a lot in this chapter. It has a haploid chromosome number of 4, so you could talk about having chromosome 1, 2, 3, 4 kind of a thing. It is a diploid organism. And then down here, humans, we have 23 pieces of DNA in our haploid number. I think the point of this slide is just to say that even within similar types of organisms, right, where we're talking about the lowly invertebrates here. Starfish have a haploid number of 18. They have lots of different pieces. Fruit flies have only a couple. If we look at Tetrahymena, which is another organism that was mentioned earlier in our Canvas course, that has a whole bunch of pieces of DNA. I don't even know what the haploid number is. I probably shouldn't have brought it up if I didn't have that number handy. But the point is that there are lots of different varieties here. Nature has found a lot of different ways to accommodate the same problem. Organisms need to have enough copies of all of their genes in order to perform their cellular activities. So how do we know? How did we know? How did we discover that all of that genetic inheritance stuff that we were able to map out and predict probabilities, whether from Mendel's work in the mid-1800s or from some of the later work done in the early 1900s, how did we know? How did we figure out that that was due to chromosomes? A lot of this came out of T.H. Morgan's lab, Thomas Hunt Morgan, working in New York in the early 1900s. He's primarily studying fruit flies, primarily studying their eye color, although his lab was really a powerhouse, massive lab studying all sorts of things. But came up with really strong proof of the chromosome theory of heredity. Also supported Mendel's principles of independent assortment and of segregation because they could see these physical pieces of DNA moving. And as a bonus, discovered sex linkage. So we're going to talk about these in the following slides. So here's Drosophila. And this is a, hopefully by now, pretty familiar looking graph. Parental strains at the top, the F1 cross strains in the middle, and then we go down to an F2 generation at the bottom. What this is showing now is a sex linkage. So we were looking at a eye color gene that's located on the X chromosome in fruit flies. Fruit flies, like humans, are a XY sex-determining organism. So when the organism has 2 X chromosomes, we call that a female. When it has an X and a Y, we call that a male. Other organisms maybe do this in a different way. We'll talk about those later, but this is how fruit flies work. So what Morgan's lab did here is to cross a red-eyed, or notice here the plus, a wild-type female, with a mutant, a white-eyed male. Now, probably you've never seen a white-eyed fruit fly. I don't know if you get fruit flies in your kitchen or anything. They happen from time to time. You've got some bananas or something that have been sitting around too long. Generally speaking, they have red eyes. Now, if you happen to see a white-eyed fly, it's noticeable. Even though their eye is super tiny, that white really stands out against the dark color of the fly's body. So these are relatively easy to see, and fruit fly labs are really, really good at identifying these different traits that we're going to talk about in this and future chapters. And they have really, really a high level of proficiency at sexing flies, being able to very quickly under a microscope tell if flies are male or female. It's sort of beyond our point to know why that is, but they can do it. So what Morgan's group did was to create or separate out a population of red-eyed females that were true breeding. So we see that homozygous dominant here, and cross them with white-eyed males. And what they got in that first generation were all red-eyed, right? And we can imagine how the Punnett square would look here. The red-eyed female would give only a red X chromosome. That'd be this one on the left in each case. The white-eyed male would be able to give either the white X or the Y chromosome. And so if we were to draw out a Punnett square here, what we would see is all of the organisms would be red. The difference here, and Morgan wouldn't necessarily have known this yet, is that the females were heterozygous with the red allele covering the white allele. The males were hemizygous, and this is a word that means they only have one copy when they normally would have two. So hemizygous for that red allele. There isn't an analogous allele here on the Y chromosome. But observationally, they were all red-eyed. Okay. Now, if we were to think about this in Mendelian terms, like sex linkage wasn't a problem here. If this male was heterozygous, just like the female, you know what we would have expected. We would have expected in the F2 generation a 3-to-1 ratio of red-eyed organisms to white-eyed organisms. So that would have been 1/4 were homozygous red, 2/4 would have been heterozygous but still red, and that last quarter remaining would have been the homozygous recessive white. So that would have been Mendelian. What Morgan's group found when they did the cross between these F1 red organisms here, this red-eyed female and this red-eyed male, when they crossed those in the F2 generation, they found that all of the females were red-eyed and half of the males were white-eyed. Now, if you just look at this and say, oh yeah, sure, that's 3 red to 1 white, that's what we'd expect, right? No, not what we'd expect, because this ratio is different between the two sexes. If this were Mendelian, we would expect 3 out of 4 females to be red and 1 out of 4 females to be white. And the same in males. 3 out of 4 males would be red, 1 out of 4 males would be white. It's not what was observed. So what was observed here is that all the females were red and half the males were red. Different ratio in the two sexes. And that's our clue that this is a sex-linked trait. Now, most sex-linked traits, when we say sex-linked, most of the time we mean X-linked, because the X chromosome is far larger than the Y chromosome. There's only a handful of genes on the Y chromosome because of its small stature. There are a few Y-linked traits that have been observed, and some in humans, but for the most part, we're talking about X linkage here. So this is the basis of what we're talking about here. How could you test that theory? Right? What would you do here to test the theory? And there are lots of different answers. I guess if I were a proper YouTuber here, I would say, put your answers in the comments. But the point is, how could we test this? Well, remember what Morgan's people would have seen. They would have been able to physically separate these red females. These would all be in one group because they can't tell the difference between the heterozygous and homozygous females. So they'd have one group here. They'd have another group here of red-eyed males, and they'd have a third group that was white-eyed males. Well, what would you do there? Many times we would talk about doing back crosses. We would talk about, breeding these out to see what happens. And we could do that. So we could look for the situation where we'd have one of those females from that red population mated with the white male. This would be considered like a test cross, mating with the recessive individual. And if that female were heterozygous, which we couldn't tell from observation, so we'd have to do this with a whole bunch of them and see what happened. But in the case that red-eyed female were a heterozygote, then we ought to be able to get a homozygous recessive female right here, the white-eyed female. She can only show up if we have a heterozygous female. So if you think about that population experiment, now we would have taken those red-eyed females, we would have separated them out, individually allowed one of them to mate with a white-eyed male, and looked at her offspring. If she was one of these red-eyed females, all of the offspring would be red-eyed, right? It would have been just like this cross here. But if she happened to be carrying that recessive allele, then we would have seen this outcome. And so that's what they did. They painstakingly separated these and mated them individually, and then they were able to get this result, which would have showed half the females being white-eyed and half the males being white-eyed. And that fit this theory that the eye color gene that they were examining was located on the sex chromosome, on the X chromosome in particular. Once we have that white-eyed female, she's pretty rare, but once we got her out of that cross we just talked about, then we can start messing around a little bit. Now we have the ability to do a test cross effectively, but with the female being the test cross partner. So now what happens? If we take a red-eyed male and we cross it with her, we would predict, based on this theory, of course, she can only give the recessive X here, so all of our males would have to be white-eyed, but all of our females would be red-eyed because they get that red allele from the male. And if we allow those to intercross together here at the F1 generation, now we should get both red and white-eyed males and females, 50-50 of each. So there are these experiments that we could design out, and if you were to draw this out in a Mendelian way, you'll see that the offspring fractions, the proportion of each kind of offspring is different, whether we're talking about an autosomal diploid gene or an X-linked situation where we can have the diploid in one sex and the hemizygote in the other sex. And so, as often happens, the big boss was satisfied here. They went on and published some pretty important papers about this stuff. Confirmed the theory that fruit fly eye color, at least for this gene, is X-linked. But then we enter the ambitious student. And I am not saying that sarcastically. What I found in my own experience in graduate school is that oftentimes students come up with these crazy, slightly off the wall kind of ideas. And even if the boss thinks maybe they're not the best idea to test, sometimes the students do it anyway. I speak from personal experience. Sometimes that pays off. What happened here is that one of Morgan's students, someone who would later become quite famous in his own respect, the last name Bridges, pushed it even further and said, well, I'm going to take these exceptional white-eyed females and I'm going to work with them further, okay? So what would you expect in the F1 generation? Well, we already saw this, right? White-eyed female, red-eyed male. Let me get back here. White-eyed female, red-eyed male. This is what we'd expect. All the females to be red-eyed, all the males to be white-eyed, right? That's what we would have expected. But where Morgan's initial experiments counted up, loads and loads and loads of flies, Bridges pushed it further. Bridges counted thousands and thousands of flies. And remember, in statistics, just because an event is really, really uncommon doesn't mean it doesn't exist. But you have to count an awful lot of instances to be able to see that uncommon event happen. And this is what Bridges did. So Bridges did observe mostly red-eyed females and white-eyed males. And when I say mostly, I mean like way mostly. But he also observed the occasional white-eyed females and red-eyed males. How could that happen? Based on what we saw here, how could we get a white-eyed female in this generation and a red-eyed male over here? Well, we can't, right? It doesn't make any sense based on these pictures. The only way that we could get a red-eyed male, for instance, is if that male inherited the X from its male parent. But it's got to inherit the Y from the male parent. So how could it inherit both? That'd be strange. And then in that case, what did it inherit from mom? Similarly, for the female, we would have to have a situation where this red chromosome didn't get inherited, and instead either just the one or maybe two white chromosomes got inherited. So that's what happened. So where Thomas Hunt Morgan's work really showed the sex linkage and the chromosome linkage, Bridges' work proved something called non-disjunction. And he had to count a lot of flies to see this happen. What this is showing us here are these parental genotypes here, right? This exceptional white-eyed female crossed with a red-eyed male. In normal cases, that female could generate a or an egg with a single white-eyed containing chromosome X, right? The male over here is going to produce a sperm with either the W plus or with the Y chromosome. And so what's shown on the left is the normal outcome, what we saw a few slides ago. We should get all red-eyed females that are heterozygous. We should get all white-eyed males that are hemizygous. But what if meiosis goes wrong in the female? And before we go too much further, I just want to mention here, we're going to talk about non-disjunction as being a female problem. It is not. I don't think there's any particular reason to think that meiosis in females is very, very different to meiosis in males as far as DNA movement is concerned. The difference here is that it is really one of magnitude. Females produce, on average, in any given average species, females produce fewer gametes than males. And so typically what we're going to be talking about is that those offspring really depend on the female gamete and their genetic characteristics. The male may very well make some non-disjoined sperm, but most sperm are not utilized in a fertilization event, and so the numbers just favor the female. So what if the female undergoes what's called non-disjunction, that for whatever reason there's a mess up in meiosis such that the X chromosomes don't separate the way they're supposed to? Well, that would give half of her eggs that are, I don't want to say half. In this rare instance, one of the eggs that she produced from that could be thought to have two X chromosomes, and the other could be thought to be empty of X chromosomes. Now, it would still have all the other chromosomes in flies, chromosomes 1, 2, and 3. But we're talking about the non-disjunction just of the X chromosome. If that happened, and it would be rare because we're talking about a cellular mistake, but if that happened, look what we would get. If that diplo X egg mated with a red sperm, we would get XXX, and what they're calling a metafemale. This in the fruit fly seems to be a lethal situation. We're going to talk about dosage compensation at the end, but in most organisms, there's a mechanism to only produce the right amount of the product of the X chromosome. And having 3 copies of that entire chromosome is usually pretty detrimental. In the fly, it seems to be lethal. So we're not seeing these ones. If we did see one of those, they'd have a red eye, and so they would just sort of be lumped in with this group on the left anyway. Well, we wouldn't know that they had 3 X's. If that diplo X egg joined with a Y-containing chromosome, we would get an XXY, which would be here now white-eyed, and it's still listed as female. And the reason for that is that although fruit flies use X and Y like humans do, we'll see at the end of the chapter, the lettering, the naming of these things is different. So fruit flies are female if they have 2 X chromosomes, but they're male if they have only 1. So it's not the presence of the Y chromosome that causes it to be male like it is in humans. It's the ratio of X chromosomes to autosomes. So here, these 2 X chromosomes would make it a female fly, but she would be white-eyed. She'd also have this little Y chromosome with whatever it encodes. In the bottom right corner, if we had a YO, so it didn't get any X, this would be the null X egg now. No X chromosomes in here. The pairing with the Y-containing sperm. It doesn't have any X-containing information. And the X chromosome is pretty important. It's one of 4 pieces of the fly DNA, so we're losing a lot of stuff. It dies. Not usually dies, always dies. But if we have that one copy from the male, even though we have nothing from the female, this XO, this thing would be able to survive. It has only one X chromosome, so it would be called a male. And that one X came from the dad, so that's red. So we'd have a red-eyed male. So we get these two quadrants would be probably lethal. As I mentioned, this first one might occasionally survive and be lumped in with the regular females that are red-eyed. But we have these two exceptional cases that formed. Non-disjunction really was the nail in the coffin for any doubters of the chromosomal theory of heredity. If Bridges' work didn't do it, if Morgan's work didn't do it, Bridges' work finding this really did drill home this concept that, yeah, the chromosomes are the important part, at least in terms of fly eye color. And of course, we know now that this is true across the board. Of course, there are sex-linked genes in humans. There are a handful of them that have been identified and studied. Hemophilia is a popular one people talk about, so that's an X-linked recessive trait. Red-green color blindness, another X-linked recessive trait, and a condition called retinitis pigmentosa, which is one of the very few Y-linked traits out there. And I sort of alluded to this. Why do we think few Y-linked disorders have been described? Well, look how small the Y chromosome is, right? It's much, much, much smaller than the X in humans, and it carries much less information. So it does have a couple important parts, but for the most part, you can survive better without a Y chromosome than you can without an X. Here is a pedigree. I mentioned in an earlier discussion about inbreeding when we talk about royal families and this sort of social pressure to marry within a royal bloodline. We can see a lot of inbreeding going on here. But that's not the point of looking at this right now. What we are seeing is that hemophilia, although it's a relatively rare disease in the population at large, it's relatively common across these royal families of we'll say Eurasia, I guess, to include Russia in there. So way up at the top, we've got some ancient ancestors here. We can see they gave rise to a couple of offspring here. Notice that this pair are siblings. So they're siblings, which means that these guys down here are more closely related than sort of general population. And so there's our first instance of inbreeding. I'll let you find the rest as we go down. But what we can see is that of their offspring, we have a couple of different colored versions here. Now in pedigrees, remember, we use color sometimes to indicate disease state. Here, the reddish color is, I'll say confirmed hemophilia. The dark red, which I guess would be like number 10 here in generation 3, that one was confirmed, so there's some historical evidence that that person had the condition. The light red over here is labeled as an evident carrier, just meaning that they had offspring later on that had the disease, and so that we can infer back that they must have been a carrier. The yellows are possible. We're not sure, but possible. And then the white is unaffected. And so what do we see here? Well, we see, firstly, an awful lot of this condition. We see it in many generations, which probably owing to some of that inbreeding early on. But what we also see here is that if we look at the offspring numbers, the probability varies between sexes. And how do we want to think about this? The key thing I want to mention to you right now is that sex determination is itself a probabilistic outcome, right? Any given sperm that fertilizes an egg will have either the X or the Y chromosome, and that's independent of any other genes and traits and chromosome pieces that it's carrying. It will have had, as a result of meiosis, either the X or the Y chromosome. And so in the Mendelian discussions in chapters 3 and 4, we didn't worry about that. We just said all the offspring were the same, right? And that's fine. But in this case, when we're talking about a gene that might be on that X or maybe Y chromosome, now we have to be concerned about whether that chromosome is even there in the first place. And so here's how that might look if we look at a particular pedigree analysis. Now, follow with me. I know there's a lot going on. It's a busy diagram. If we go up here at the top and we're going to say, let's look at individual 4 here to the right and generation 4. Okay, what's the chance that that individual is going to be colorblind? Okay, now colorblind is a sex-linked trait. It's recessive. It's on the X chromosome. And so what we're going to say here is, well, we're not sure about this person, but let's look at the family. Let's look at the parents. Okay, so now we see that this number 4 is a known carrier, female, and this individual is a known normal, I hate the word normal, but a non-carrier, male. When they mate together, what's going to happen down here? Okay, so let's think about this. What's the chance that they're colorblind? So firstly, in order for them to be colorblind, they would have to inherit that allele from a parent. Okay, so that mutant allele has to be transmitted in the first place. That'd be a 1 out of 2 chance if this parent was a carrier. And they would have to have the Y chromosome transmitted from the father. And now why the Y chromosome? Because in that case, the offspring would be hemizygous, and so that one copy of that mutant allele would be sufficient to give us the condition. So I want you to be careful about your probabilities here. What's the chance that this individual would be colorblind? It's 1 out of 4. And essentially that's 1/2 chance that they're male, that they've inherited the Y chromosome. And anytime we say and, remember we multiply. And a 1/2 chance of inheriting the mutant allele from mom. So 1/2 times 1/2 is 1/4. However, if the problem said, what's the probability of having a colorblind boy? Okay, a colorblind boy child. Now we don't have to worry about this, the 1/2 for the Y chromosome. That's already been done by saying we're only looking at the males. So the answer would be 1/2. So if we looked at all of our individuals, half of our male offspring would be, or we'd have a 50% chance of a male offspring being colorblind. We'd have a 0% chance of a female offspring being colorblind, although she could be a carrier. But altogether, combining those two probabilities for all children, we'd say 1/4 chance of any child being colorblind. This is what you would say to a family if you were doing, say, genetic counseling. You'd say, okay, if we know that mom's dad up here, grandpa, was colorblind, all right, then we know that mom is a carrier. If mom's mating partner out here doesn't have that disease, no family history, nothing like that, we'd have exactly this situation. And so if they came to our genetic testing clinic, right, and they said, you know, mom said, I have this history of colorblindness in my family. What's the chance of my offspring, my kid, being colorblind? We'd say 1 in 4. It won't happen to a female child. It could only happen to a male child, but 1 in 4 overall probability. Now on the left, these sort of warmer colors over here, we're going to look at 2 generations down. Now we have a little bit of uncertainty, okay? If we look at the bottom, we're going to say, in order for this bottom individual, 5-1, to be colorblind, they have to be male, okay? Because their parent isn't homozygous, right? So they have to be male. So we get that hemizygous condition. That's a 1/2 chance. Then also, they'd have to inherit that mutant allele from their female parent. That's a 1/2 chance if she's a carrier. But the chance of her having been a carrier in the first place is a 1/2 chance. And so if we don't know her genetic status, then we can say, all right, now we're a little further away. We have to say there's a 1/2 chance that you were a carrier in the first place. If you were, you would have to donate to your offspring the mutant allele. That's another 1/2 chance. And your partner would have to donate the Y chromosome. That's another 1/2 chance. So based on the information we know on the left side of this pedigree, we would say the chance of individual 5-1 being colorblind is 1 in 8. Again, it would have to be a male, but a 1 in 8 chance overall. So when you approach these probability sorts of questions, do it step by step by step, right? And I like to at least talk out loud. Okay, what has to be required here? Well, the allele has to go from this parent to this parent. It's a 50-50 shot, 1/2. By the way, fractions are way easier than decimals here, so try to get in the habit of using fractions. Moving that mutant allele from 3-2 here down to 4-2, that's a 1/2 chance. Once that allele's there, that'd be a 1/2 chance of transmitting it further, and the father would have to transmit the male chromosome, so we get a 1 in 8 chance, 1/2 times 1/2 times 1/2. Practice those problems, okay? And please be very careful about the language. Again, let me just pop forward for 1 second to the textbook. If we look at a problem here from the back of the textbook, problem 5.9 gives us a setup that we could draw a pedigree. So a man with X-linked color blindness marries a woman with no history of color blindness in her family. We can draw those out. The daughter of this couple, so draw her. She marries a normal man. You can draw him. Now we have 2 generations. Their daughter now also marries a normal man. There's a 3rd generation. What's the chance that this last couple will have a child with color blindness? So you're essentially building a pedigree very similar to the one that we just saw. And they're asking, what's the chance that this last couple will have a child, any child, with color blindness? So you're going to have to include that sex determination factor in there as well. If this question said, what's the chance of having a colorblind boy, or what's the chance of having a colorblind girl, then you would need to do those separately. This problem goes on further. If this couple already has had a child with color blindness, what's the chance that their next child would be colorblind? And before you very quickly say, well, it doesn't matter, one's the same as the other, they're independent. Having an offspring with color blindness tells us something about the parent's genotype. So we get from that possible carrier kind of a setup to a known carrier, and that changes your probabilities a little. So 5.9 is a great problem to look at. The answer's in the book. I have video walkthroughs in the Canvas module about 5.10 and 5.12, just to see, a couple of examples of how to work through some things so you can practice those. So back to the slideshow. Just going to finish up with following up on what I said earlier. In many organisms, we're going to talk about the sex chromosomes determining male and female phenotypes. That's why they're called sex chromosomes, but in different ways. So in fruit flies and humans, I mentioned they follow the same system, although star there because they follow it in different ways. Humans, the Y chromosome is the determining factor. Having a Y chromosome makes a person biologically male. In fruit flies, it's this ratio. So having 1 X makes you biologically male. Having 2 X's makes you biologically female. But in both cases here, we would say the heterogametic, or having 2 different sex chromosomes. The heterogametic sex is the male. The homogametic sex, 2 of the same, is the female. In birds and butterflies and some reptiles, there's a difference there. It flip-flops. So the males have 2 of the same chromosomes. Females have 2 different ones. In order to make that more clear, instead of using X and Y, we use the neighboring letters Z and W. So a male bird, for instance, would be a ZZ sex chromosome configuration. Honeybees do this a little bit differently. Males are haploid and females are diploid. There's a whole thing about bee sex determination and social structure and stuff that you can take a look at if you're interested. Here's that fruit fly thing, because this whole chapter focuses in on Morgan and Bridges and fruit flies. Here's the situation. So normal males and females would be here. So having a single X chromosome or two X chromosomes in an otherwise diploid organism gives us these ratios. That's our male and female, like normal. When we start talking about non-disjunction and sort of weird chromosomal abnormalities that might happen in a rare kind of a scenario, we could imagine ending up with 3 or 4. So 3 X chromosomes in a normal, otherwise normal cell would be our metafemale. This is the one that chart mentioned usually dies. So it's pretty stressful for the cell. Having 4 X chromosomes, but now more autosomes, gives us a ratio that's a little lower, so we can get metafemales that are a little bit different. In extreme cases, we can get some very unusual chromosomal arrangements here, and we can see sort of the outcome. So standard fruit fly males and females are up here, and then this is just showing us that there's a lot of other stuff that can happen. In humans, here's the sort of setup. Both sexes start with the same embryonic gonadal tissue shown here. If we have 2 X chromosomes, we follow what could be considered a default pathway that gonadal tissue will turn into ovaries. Those ovaries then start to produce female sex hormones that cause secondary sexual characteristics to develop. In the presence of a Y chromosome, and more specifically this SRY region, which stands for sex determining region on the Y chromosome, SRY, that gene produces testis determining factor, which then causes this tissue to, instead of becoming an ovary, to become a testis. That testis then produces male sex hormones, predominantly testosterone, and then that causes secondary male sexual characteristics to develop. Now, obviously, there's a lot of moving parts here. So if we wanted to get into a discussion of different levels of sex hormones and impacts on the body and all those secondary characteristics, we could certainly do that. But from a very basic standpoint, this is why, for humans, the presence of a Y chromosome leads to generally male sexual characteristics. Now, importantly, that is one gene on the Y chromosome that's critically important. So what if we had some weird genetic thing that deleted or inactivated that region, right? We talked about mutations and stuff. What if we had a hypomorphic or a null allele in that gene? Well, now an organism that was XY from a chromosomal standpoint would not have that key region, would not produce the testis determining factor, would not generate a testis, would not generate testosterone. They would develop as a female, even though they have most of the Y chromosome. So on a karyotype, on a genotype, you'd say, well, this individual is an XY individual, but all of their measurable, observable characteristics would be those of a female. On the other hand, if we had some sort of a gene transfer event that moved from this normal pairing, moved that region of the Y chromosome over to the X chromosome. This could happen during those cellular processes where the X and Y are homologously paired, right? So mitosis or meiosis. If we had a rare event where this little bit of the Y chromosome was moved onto an X and then that X got inherited into an individual, now this individual would have 2 X chromosomes, so chromosomally they would look female, but they'd have the SRY region. They would produce early on that testis determining factor. Their embryonic gonad would turn into a testis, and that testis would produce testosterone, forming male secondary sex characteristics. So why is this slide here? I think it's to remind us that when we say words like normal, when I talked about fruit flies having that normal ratio of 1X to 2 autosomes or 2X's to 2 autosomes, there's lots of other stuff that can happen. And just like in the case of Morgan's work compared to Bridges' work, if we look at a handful of things, we will see, a result. But if we look at many, many, many times more, we'll see those rare results happening. Having a deletion or an inactivation specifically in that SRY region is pretty rare, right? Of all of the pieces of the genome, we'd have to inactivate specifically that little bit. It's not relatively likely. But if we look hard enough, I bet we find this in a fair number of people. Those people would have grown up to be females in an era where we didn't have the ability to do genotyping, or 23andMe wasn't a company, which maybe it isn't anymore. I don't know. Um, if we didn't have the ability to look, these people would have just developed as females. They would have grown up as little girls who became young women, who became women, and maybe they wouldn't be totally fertile. Oftentimes, when we have chromosomal abnormalities, we get a decrease in fertility. So maybe they didn't have their own children and the mutation ends with them. Or maybe they did have their own children and it gets passed on. We wouldn't ever know. Now, in this era where we can very easily do chromosome spreads and analyses, we can do genetic sequencing and testing, we're finding more of these things that happen. And that's true, we'll see in the next chapter, all sorts of different chromosomal, what the book calls abnormalities, but differences from what we see in most of the population. Here's sort of an interesting one that is just kind of fun for the biochemist in me to talk about. On the left, we see a normal male, everything normal, the way we think it ought to work. And what this is showing us is that SRY region I mentioned produces, as I said, testis determining factor, which causes the gonad tissue to turn into a testis, and then it produces testosterone. Okay, we covered that. The part I didn't tell you is that the receptor for that testosterone, that receptor is encoded by a gene that's on the X chromosome. So practically, in order to be a quote unquote normal male, I need both an X and a Y. I need the Y to produce the thing that's ultimately going to cause testosterone to be produced. And I need the X to produce the receptor for that testosterone to be able to signal differentiation in other tissues. I need both. So we talked about on this slide what happens if I'm missing the SRY gene. Well, what happens if I'm missing the receptor or it's mutated? This is kind of neat. Now I produce SRY, so I produce a testis, I produce testosterone, but that testosterone can't signal anything. The signal's there, but it's not being received. So what does that mean? Well, that means I go down that default pathway of sexual characteristics. Although I have a testis and although I produce testosterone, I'm not going to develop a lot of the typically male secondary sexual characteristics. So this is a concept called testicular feminization. It's kind of a neat condition to think about. This individual would have testes rather than ovaries, but in many other scenarios, they would look more female in terms of body shape and observable characteristics. Kind of a neat, again, rare instance to talk about for human sexuality. This is just showing us the ZW system. I don't know if this is really necessary, but the key point here is just that the female is the heterogametic sex now. So she has two different chromosomes, whereas the male has two of the same. And you can see how they mix together. I don't know what these yellow lines are all about. It's pretty crazy. But this is just a normal cross. This is the haplodiplo system in honeybees. For instance, a diploid queen bee can lay an egg that stays unfertilized, and that becomes a haploid male. And then, alternately, she could have an egg that gets fertilized, and then that would produce a diploid again, which would be the female. So there aren't two different chromosomes in this system. It's the same sex chromosome, but having two of them makes you a female. Having one of them makes you a male. And in fact, not two of them, it's the entire genome. So the key thing here is that all eggs develop whether they're fertilized or not. It's kind of a neat system, different to most of the other ones we've talked about. Likewise, just one more little concept here at the end called dosage compensation. I mentioned in passing earlier that because there are a lot of genes on the X chromosome, that we need to control the amount of expression of those genes. If you think about, say, in humans, if a male has one X chromosome and a female has two, without any sort of mechanism of control, the female would produce, on average, twice as much of all of those X-linked gene products as the male, and that probably wouldn't be good. There are 3 ways to adjust for that in nature. So the one that humans do is number 2 here. One of the female X chromosomes gets inactivated. So females become functionally hemizygous by having one of their X chromosomes inactivated. In Drosophila, they do it the other way around. They get hyperactivation of the male X chromosome, right? The male only has one X chromosome. That thing gets turned up in its expression level, so it produces twice as much as each of the female ones. So at the end of the day, the chromosome expression is normalized. And in roundworms and other species, there's a hypoactivation. So the point of putting this slide up for you is just to say the key concept here is that males and females have to have similar levels of expression. We can do that in several different ways, and nature has done it in several different ways. The last super cool thing here at the end is mosaicism. Mosaicism is a concept where an embryo could have its X inactivated differently in different cells. And it would get us a little too deep in the weeds to talk about how X inactivation works, but suffice to say that you can end up in a scenario where at an early stage in the organism's development, one cell might inactivate X chromosome number 1, and the other cell might inactivate X chromosome number 2. And then all the descendants from those cells would have that same inactivation carried on. And in this case, what they're showing us is that they're trying to color code these. Okay, so in some of the cells, we might end up with a dark fur pigment activated, and in the other cells, we might have a light fur pigment, and we might end up with this sort of mottled looking little cat. This is called mosaicism. It can happen in all sorts of different ways. There are pigment conditions that are thought to result from mosaicism in different cells. Heterochromia here, having 2 different colors of eyes, is thought to result from that. There are links at the bottom of the slide here that you can follow to talk about those. All right, so I would encourage you as you finish up in this chapter to really focus in on those Punnett squares. Really think about using your Punnett squares in, I'm going to go real quick back here, sorry, click. Something like, well, I guess I'll put this one up, sorry. Something like this. Think about having your inputs to the Punnett square being a little bit more graphical. So when you do the sex-linked Punnett squares, you're typically going to show the letter X or Y. And if it's an X, you'll use a superscript to indicate what the allele is, whether it's dominant or recessive, whatever it might be. And then in the Punnett square, when you look at those crosses, you're both interpreting the phenotype in terms of dominance and recessiveness, but also the sex based on XX and XY content. And remember, fruit flies and humans use the same XY rules for sex determination. So different reasons, but the same output. So practice with these, work through some problems at the end of the chapter. And as always, reach out to me if you have any questions. I'm happy to work through things for you. So with that, keep up the great work and have a great day.